Only few people are listening, I think. Nobody is responding. Statistics or probability. Okay. Yeah. So we use rule-based inferences to calculate among the 5 experiments, considering all the 5 experiments, how we can calculate or choose the best 2 molecules, right? So we use a rule-based inferences by doing probabilistic scoring. So we have data from in silico, in vitro, and in vivo, and then we have to prioritize which property or which experiment is more important to us for make decisions. But I said all 5 has to be still considered. But among the 5, which one is more important? So which one is more important based upon your understanding of relevance? And second, let's say one experiment is very important, but the results are unreliable, lot of errors, too much standard deviation. Don't consider that as an important experimental data because you have a lot of errors there. When you consider data with more error, your prediction also will be bad, right? And finally, you make selection based upon understanding quality and diversity. This is a normal scenario what we do. You have set of molecules, you try to do bioassay studies, and then you select some compounds which is having good activity. And then you do some absorption rate studies and metabolic stability studies. Each time you will remove some compounds which are not satisfying the experimental threshold. And then you expect that you will get a very good compound at the end. Unfortunately, no. Even though your compound is very absorption rate might be less and it is being thrown out. So you are missing that compound. Your compound might be at the end not reaching the last stage because you are ending up with some non-promising compounds. I'm sure the faculties would be agreeing to what I'm trying to say here based on your past experiences in research. You end up with some moderately active compounds, not very good when compared to standards or anything in all aspects. No, I'm not talking only about potency. I'm talking about in all aspects, right? So you have to be very cautious in what kind of methodology or approaches you're going to choose. So I would like to stop here. I'm not moving forward much. This all will be too much for you, but I'll just say what is ADME. So ADME is that A is for absorption, right? So we are trying to understand whether my drug molecule can be absorbed by this intestine, small intestine of humans. And then when you talk about D, it is distribution, where I'm trying to see whether my drug, where will it go, where it could distribute it after getting absorbed. And then M is for metabolism, to understand whether my drug compound can remain intact or whether it form metabolized or whether it is unstable. And E is for excretion, to understand whether it is going to accumulate or whether it can be released properly outside the body. Now you might think, how can we use ML or computational approaches in order to calculate ADME properties? That is just because you can correlate your molecular structure property, that is called physiochemical properties, with respect to biological activity or ADME properties. For example, if you look at the distribution here, log P, pKa, molecular weight, hydrogen bond, polar surface area, and transporters of the molecular properties are correlated with the distribution. That's how the structural features can be correlated with ADME property. Similarly, toxicity also and different other parameters. Different ADME properties that could be also predicted based upon structure activity relationship model. Then you can also predict toxicology or environmental and developmental toxicities also could be predicted based upon structure activity relationship. Human toxicity endpoints also can be predicted for 43 human toxicity endpoints, which is again not on animals, but on humans. These are all based upon clinical trial data. And then you can also check with whether which region of your compound is regioselective for P450. And also you can design new molecules based upon specific property. For example, let me tell you that a small example. Let's imagine that compound A and B. A is very active, like very highly potent. But when we did toxicity studies, we understood A is also toxic. right? Now we have another compound called B. B is moderately active, not that much active, right? But B is very much non-toxic. Now, question two to all of you. Do you consider A or B for your optimization or making changes and other things? So, what kind of strategy that you will follow to consider whether it is A or B? And you have to tell me the answer also. I mean, reason also. Why do you choose A or why do you choose B? As I told you, A means it is very active but still toxic. B means it is moderately active but non-toxic. Anybody can answer, including faculties. Well, what is the strategy that you're going to take?
Sir, B, sir.
Why, Mukesh?
Sir, it is moderately active means uh, uh, we can give some other compounds to activate it faster. But if it is toxicity means it will be affect us, no, sir. So that we
Okay,
can prefer non-toxicity.
I can accept that answer. But what is your first priority to choose? So you told me that you're going to make modifications or you're going to add some other molecule to activate it. That is absolutely fine, agreeable. But now, if I'm asking you to, what is your first priority, A or B, to do that same process?
Sir, by taking risk, sir, it is also toxic. Once we can take risk,
Okay,
we can
so
take.
it is always a risk. Drug discovery is a risk, and you have to expect 99% failure when compared to success. So the rule of thumb is you have to consider A first. That is a priority because A is
So,
having more talk. Yeah,
so even we can reduce the toxicity level, no, sir.
exactly. That's where I'm heading to. So
Okay,
you
okay,
have
sir.
to consider A, which is more active because activity is more important for us. That is the most topmost priority for us. So we need to see whether which compound is having better activity or better affinity, and then try to minimize the toxicity by making modifications or by adding some other additives or excipients as Mukesh suggested. But when you come to choosing the best molecule A or B, we always end up with choosing A. If A, after making modification, if the A did not work, then only we go to B in order to improve its activity. Because we already know A is very active. So we have to work on top of that first because that is our topmost priority. Activity is very important. And there is also another answer from Tarani that A can be used in small quantities to regulate the toxicity levels. If B is used the large quantities, that will be if needed. So A is the first priority. That's a good perspective, Tarani. See, that is what we have to look at. I know everybody would have read about DRDO's compound 2DG. How many of you know the history of that compound? Have you ever tried to read a little bit more about that? I'm not sure. I have gone through it and I did a podcast also for that. 1958, that's where this compound was first synthesized. 1958. So now you will ask me, sir, all media was telling DRDO has developed. No, they have not developed. 1958, this compound was first synthesized. In 1958 till 1978, they were being studied on anti-cancer studies. I'm trying to say something what Tharani said. So on a lower concentration, the the result of the anti-cancer studies was not that great. When they increased the concentration, they had a better response, but on a prolonged usage of 2DG, it was very toxic. So now you understand what I'm trying to say. So in 1978, they closed that research because it's a good compound, but further optimizations are required because it is toxic for a prolonged usage. In 2008 and 2009, from INMAS DRDO, the professor wrote a review. In his review itself, he mentioned about all this 1958, 1978 papers and all. There he didn't mention anything about COVID, he mentioned about cancer research. But then recently, many people, not only him, but many others also claimed about using it for antiviral thing as a precautionary complementary or integrated approach of medicine and nothing like a direct approach. So what I'm trying to say is, when you say when you're trying to do it for small quantities with a compound, the mode of action or the activity might be also less. And when you increase the quantity, that's where it is toxic. Your way of thinking is absolutely right. So the dosage form is very, very much important when you talk about toxicity as well as activity, right? Great ideas. That's really welcoming. So these are some of the research outcomes, some of the selected papers I have chosen where you could go through on some of the reviews to understand how these, whatever things I have discussed today is being applied. So that's all from me. These you can get all the slides from SlideShare. So you can get much more slides, including chemical biology databases, then natural product databases, then how to identify targets, how to validate it, and many more others. And we do take interns for master students, and we take only 2 per year after screening process. The internship is completely free. We don't charge anything, but there will be interview and project proposal coming from your end. And I have been uploading a lot of demo videos for drug discovery related, especially docking, QSAR, molecular dynamics in my YouTube channel. So this is the link that is for that particular MD simulation studies. So that's all from me for today. If you have any questions or disagreements or something that you want to comment, I'm happy to take it.
Sir.
Yeah.
சார் நீங்க சொன்னீங்க வந்து ப்ரிஃபர் கெமிஸ்ட்ரியில ப்ரிஃபர் பண்றது ஏன்னு சொன்னீங்களா சார்? வந்து டாக்ஸிசிட்டியா இருந்தாலும் அது ஆக்டிவா இருக்குன்னா அதெல்லாம் ப்ரிஃபர் பண்ணுவாங்கன்னு.
ஆமா
சார். ஆமா சார். அப்படி பார்த்தா நம்மளுக்கு ஒர்க் ஒரு டபுள் ஒர்க் ஆகும்ல சார்? ஃபர்ஸ்ட் நம்ம வந்து அந்த காம்போனன்ட்டை அது கூட இது பண்ணி அதை வந்து நம்ம என்ன வேகமா பண்ணாலும் பின்னாடி அதுல இருந்து நம்ம டாக்ஸிசிட்டி ரிலீஸ் பண்ணி அதுக்கப்புறம் தான் நம்மளால அது அந்த ட்ரக் வந்து என்ன ஒர்க் பண்ணுது, அது வந்து எங்க போய் பைண்ட் ஆகுது, அதெல்லாம் பார்க்க முடியும்ல சார்? ஆனா நம்மளுக்கு வந்து டைம் கன்சியூமன்ஸ் தான் சார் பீல் ஆகும் எப்படி இருந்தாலும். ஏன்னா டைம் மட்டும் தான் கன்சியூம் ஆகும், வந்து அது மெதுவா எடுக்கும். ஆனா வந்து அது கரெக்ட், அது வந்து நம்ம டாக்ஸிசிட்டி வெளியே எடுக்க தேவையில்லை. அதுக்கப்புறம் வந்து நம்மளுக்கு அது டக்குன்னு வந்து டாக்ஸிசிட்டி எடுக்கிற டைம் நம்ம இதுக்கு ஆயிக்கும். பிளஸ் வந்து அதுக்கான ரேட் காஸ்ட் கம்மியா இருக்கும். ஏன்னா டாக்ஸிசிட்டி எடுக்கணும்னா நம்மளுக்கு அதுக்கும் ஒரு அதுக்கும் வந்து நம்மளுக்கு மெட்டீரியல்ஸ் தேவையில்லை சார்.
I understand, Mukesh. Really encouraging with your type of questions and thoughts. You're very true, but unfortunately, drug discovery is like that. You have to take risk. It takes time, and you have to do trial and error. Increase a specific concentration, check everything. Then reduce a concentration, check everything. When you're reducing the concentration, if activity is less and toxicity is also less, then what is the point of calling it as a drug? When you increase the concentration, activity is very good, but still it is toxic. So still you have to do some optimization. There is a risk factor. You have to spend more time, do more research, synthesize it, again redo the things again for testing. That is the process of drug discovery. That is why it is said it will take a minimum of 6 to 12 years of time frame for the market, just because of this iterations of optimization. So for drug discovery, optimization is the major part which takes more time for testing and other things. Other in the pre-clinical phase studies எல்லாமே ஒரு காம்பவுண்ட்ல பண்ணி வருவாங்க, ஒன்னோ ரெண்டோ இல்லட்ட 10 காம்பவுண்ட்ல பண்ணுவாங்க. மறுபடியும் இதுல எல்லாம் ஃபெயில் ஆச்சுன்னா மறுபடியும் ஆப்டிமைஸ்
சார். அப்படி பார்த்தா நம்ம டைம் கன்சியூமிங் வந்து பி ரொம்ப கம்மியா இருக்குமா சார்? ஏன்னா நம்ம வந்து அதை ஆட் பண்ணி விட்டுட்டு நம்ம வந்து மத்த ஒரு ட்ரக்குக்காக டிசைன் பண்ணா கூட இது வந்து ஸ்லோவா டைம் எடுத்து நம்மளுக்கு ஆனா வந்து கரெக்டா ஆயிரும்ல சார் எப்படி இருந்தாலும். அதுக்கு பி தான சார் ப்ரிஃபர் பண்ணுவாங்க.
I'll tell you an example.
பீலி ரிஸ்க் இருக்கு சார். பீலி ரிஸ்க் இல்லாம இல்லைங்க. ஏன்னா பீலியும் என்ன வேணாலும் ஆகலாம் அப்படிங்கிற நம்மளுக்கு ஒரு ரிஸ்க் இருக்கு. ஆனா அதுல வந்து ரொம்ப ரொம்ப ஜாஸ்தியான ரிஸ்க். நம்ம வந்து நம்ம எடுக்கிறோம். ஆனா ரொம்ப ஜாஸ்தியா எடுக்கிறோங்கும் போது அது வந்து நம்மளுக்கே ஒரு விதமான ஒரு பயத்தை உண்டாக்கிரும். பிளஸ் வந்து நம்ம
No, that is all a part. Let's look into the research part. So, இப்ப நீங்க B எடுக்கிறீங்கன்னா, let's consider corona. Okay, கொரோனா வைரஸுக்கு நம்ம ஒரு B மாதிரி ஒரு காம்பவுண்ட கொடுக்குறோம். மெதுவா ஆக்ட் பண்ணா போதுமா? மெதுவா ஆக்ட் பண்ணா it is again risk to your life. So, the time of action is very important. So there is a thing that you have to consider: the retention time and the activity time, all those half-life of protein, all this has to be considered. So B தான் எப்பவுமே first priority ன்னு சொல்ல முடியாது. So there, as I told you, B also can be considered. ஆனா அங்க activity வந்து நமக்கு நல்லதில்லை. So we have to first prefer a compound which is more active. I did not say you shouldn't consider B. You can consider B also, but after considering
sir A. One question, sir.
Yes.
Only one question, sir. Last question.
Yes.
சார் இப்போ வந்து நம்ம ஏ மாதிரி நம்ம ஃபர்ஸ்ட் ஏ வந்து கொரோனாக்கு வந்து பி ஆப்ஷன் எடுத்தோம்னா நம்ம வீட்டுக்குள்ளேயே மாஸ்க் போட்டு
don't tell me all those things.
ஆனா
Don't tell me all those things. Don't be specific to corona. Don't be specific to corona. Just tell me only about drug discovery. Only drug discovery. Corona பத்தி பேச வேணாம். அது மாஸ்க் அதுக்கு வேற விஷயங்கள். I'm just focusing only on drug discovery. So tell me on that.
இல்ல சார், நான் வந்து நீங்க சொன்னதுக்கு நம்ம ஏ இப்ப எடுத்தனால தானே சார் பிளாக் ஃபங்கஸ், ஒயிட் ஃபங்கஸ், yellow ஃபங்கஸ் வரைக்கும் வந்திருக்கு.
Let's don't discuss about that. Let's don't discuss about that. I am talking normally on a drug discovery aspects. I just took corona only for the time of action because we need immediate cure. That's what. Yeah.
Okay, sir.
Because எல்லா ட்ரக்குக்குமே சைடு எஃபெக்ட்ஸ் இருக்கு. எல்லா ட்ரக்குமே டாக்ஸிசிட்டி சைட்ஸ் இருக்கு. So if we start discussing about that, it will be a never ending discussion. So only concept here is that when you're trying to choose which compound for your optimization or which compound to be considered for modifications, that is where we are looking at A and B. ஆனா அது A and B வந்து கண்டிப்பா டிஃபர் ஆகும் based upon specific diseases. நீங்க சொன்னது ரொம்ப கரெக்டான வேலிடான பாயிண்ட் தான். இப்போ ஒரு வியாதி வந்து அதுக்கு மேல வேறொரு வியாதி வருது, let it be anything based upon a drug or action, let it be anything. அதோட டாக்ஸிக் எஃபெக்ட்ஸா இருக்கலாம், but it depends upon disease to disease. That we cannot generalize it here, right? So that is what my point is. Great. So any other questions?
This session is now open for queries and students can interact with the guest speaker now.
I think only Mukesh, I remember Mukesh, Tarani and Ramya, they are interacting. Apart from others, they're all keeping silent. But many of them responded for my yes and no answer. That is also great. How about others? Do you all know what is the purpose of conducting these sessions, seminars, conferences? Can anybody tell me? The funny answer would be to have a good lunch and dinner, right? But what is the main purpose? Can anyone tell me what is the main purpose of conferences, seminar, and sessions? From external people, not internal your faculty. I'm talking about why a student should attend conference, seminars, and sessions.
Knowledge sharing, sir.
Okay. Anything else?
To exploit information from others.
Okay. Any others? To gain and learn
also
about
to
all new
gain
things.
knowledge, sir.
Okay.
To get a relationship with the practical oriented peoples.
Exactly, sir. Dr. Chidambaram sir said it rightly. The main purpose is not to get new knowledge. Of course, we want to get new insights from people who are sharing their work. So when I was an undergrad or a master's, when I was sitting in front of a lot of conferences, people will come and talk about their work. I did not understand a single slide from there, right? I never understood any single slide, to be very honest with you. Because I am still doing a UG, he was talking about some very advanced work. How can I understand? But as Dr. Chidambaram rightly mentioned, these conferences are all for networking. The main purpose is networking. So what I did was, I didn't understand anyway his talk, nothing I understood. But then I went to his website and understood he's working on specific areas. After his talk, I take an opportunity to discuss with him. Sir, I saw that you're working on that. I'm also interested in that area. Even though I don't know anything about his work, I didn't understand anything about his work. I talked to him. I'm very much interested to learn about this and can I join as an intern at your place? So we made a relationship there by talking about his work. That networking is the only purpose for a student to go for a conference. Now you will say, sir, I need certificate for that. No companies will ask you certificate for conference attendance or participation. They will only look at certificates if you have got a first like place in poster presentation or oral presentation. Yes, then they will look at it. For participation, they never look at it and they don't require it and they never count it. So that is the point of thing that you are look at. When you network, no, the money you would have paid for 500 or 300 or even 10,000 rupees for the conference is full-fledged because you're going to take a next step in your career or next step in your career research. I'll tell you an example. I was working in Bangalore back in 2007. At that time, I came to Cochin to give a talk. And during that time, a company called BASF, I don't know how many of you know that company called BASF, came to also for the same conference to give a talk. They saw my talk. After that, they interacted with me. And during the interactions, everything went well about the work and all things worked. And next day morning, they said they want to meet me. And they met me and they told me, are you looking for a career change? We would like to hire you, right? Just because of that conference and because of the network that happened. Yes, I took up the job. I went to Mumbai. So that conference was that networking helped me to get a job. That is the benefit. So if you're keeping silent, oh, am I going to say something wrong or bad? Now you might be also thought that why Mukesh is asking too many questions. No, that is the right thing he has done because he will never get an opportunity to interact with me later like this. He might contact me through email, but now you shouldn't miss that opportunity. You shouldn't feel that, why he took so much time for discussion, blah, blah, blah. No, nothing. That is the golden opportunity that he can utilize. That's the way every student should be while making use of these sessions. It's not, of course, information, knowledge gaining. You can also do it from the web, right, nowadays. Earlier days, until someone comes and share it, we don't have. But nowadays, who cares? Everything is available on YouTube. Everything is recorded and kept on the archives. Everything is available on the papers, reviews. You have plenty of resources for that. So the main agenda for all these things is it's very important for students, especially to interact. In US, when I was doing my PhD, I spent around $1,200 to go for a specific meeting in order to meet a professor because I wrote several emails, he never replied. But after that, we had a very good relation. We published papers, we did patent, and all those things happened, many things. So networking is something very, very important for these kinds of expertise. Now, when you look at faculties, faculties do get new ideas, or they might be thinking about their research work in a different perspective. When external people come in and they give a little bit of deeper knowledge about what they worked and how they worked when they discuss, the faculties also will get a new perspectives on that. See, I cannot say everybody is an expert in every area. We are learning every day. So new perspectives will help us to come up and innovate or discover new methodologies. So networking is always helpful for everybody in respective domains of their expertise. So make use of it, not only now, hereafter, whenever you have any sessions with external people. Yeah.
Thank you, sir. It's a very nice presentation.
Thank you, sir. Thank you.
If any queries, they can get clear with Ramya.
Yes, sir.
Hmm.
Students can interact with the guest speaker if you have any queries.
If it's not so, you can wind up, Ramya. You can finish
Okay,
it
sir.
off.
Okay, sir.
Thank you. Thank you. Thank you.
Thank you, sir, for the insightful speech. To formally close today's meet, may I call Ms. S. Yogapriya from 2nd BSc Biotechnology to deliver the vote of thanks.
Good afternoon to all. I deem it a great honor and privilege to prepare the vote of thanks on this memorable occasion. Let me first of all start by giving glory to the Almighty God for making today's occasion a resounding success. First and foremost, I thank our special guest, Dr. Girina G. Pillai, Chief Scientific Officer, Zastra Innovations, for his useful lecture on role on ADME and toxicity in drug candidate selection and AI approach, who, despite his busy schedule, has found time to grace this occasion. And I also express my heartfelt thanks to our principal, Professor Dr. V. Rajendran sir, in his absence. Next, I thank our head of the department, Dr. P. Chidambara Rajan sir, for his good guidance and encouragement in his efforts. I also thank all other staff members without whom the session would be incomplete. Last but not least, I thank the other students for their cooperation. Thank you.
Students are
Thank you.
requested to fill the feedback form, which is in the chat box. Thank you, sir.
Okay. Thank you so much.
Thank
Stay
you, sir.
safe.
Thank you,
Bye.
Mr.
Thank you.
Thank you so much, sir. Thank you, sir.